Okay, so next up we have Michael Evers, who, as Holly said, is new to AIM. So Michael received his PhD in biomedical engineering from the University of Lübeck in Germany. And then he did his postdoc in biomedical engineering at Harvard Medical School. And he's currently in the Department of Dermatology at the Massachusetts General Hospital at the Harvard Medical School. And so he focuses on basic and translational research, looking for novel diagnostic methods and treatment devices. So why don't you go ahead and share your screen, Michael, and I'll make sure you're all set up.
Okay, there's the endless loop. That works. Okay, can you see my screen?
Yes, I can see you and hear you. You're ready to go.
Thanks so much. First of all, thanks so much for the invitation. This workshop is really great. I attended in 2017 while I was doing my PhD thesis on this topic I will present today. And it's just great talking to people and really getting to know them, and also like feeling comfortable that what you're seeing with FLIM is actually really happening, because especially like with these techniques, it's very challenging sometimes, especially if you're new, to really be confident in your data. And now I feel honored to present my PhD results over here. So I would like to start with my title. It's we're looking at Adipocytes, and we are using two-photon excited NADH FLIM. So why are we interested in, let me get my little pointer here. Why are we interested in fat at all? Why do we want to look at fat? We are interested in it because fat depots are not all the same. There are actually 3 different fat depots in the human body. And first of all, I would like to talk about the white fat, which is the fat probably most of you know. It's like the fat we have in the body to store energy. And as you can see, we have a large lipid droplet. And I will go into this in more detail later, but it does not have UCP1 expression, which is uncoupling protein 1. And then we also have brown fat, which is used to dissipate energy. You can see it has small lipid droplets and a lot of mitochondria, and it has a high UCP1 expression. And then there was another type of fat that was discovered in 2012. It's beige fat, and it's a mix between white and brown. And you can also see it has a high UCP1 expression. So if we analyze these 3 types of fat, we would basically say white fat is bad fat. You don't want that. You should avoid accumulating too much of it. And then brown and beige fat is actually fat that we want. So why do we want beige and brown fat, and why do we have 3 types of fat in the human body? When humans are born, we are actually not able to shiver yet. So if a baby gets cold, it activates that brown fat, it burns energy, and the baby gets warm. And you can see here in this comic, the brown fat is around the neck area. Once we get older and we learn the ability to shiver, people thought adults lose all of their brown fat depots. However, there was a paper in 2009 that showed if you have a normal adult human lean person and put that person into a PET scan after injecting a radioactive glucose tracer, these areas where the brown fat is supposed to be don't take up the glucose, so there's no increased activity. However, if you put that same person into a cold room so that it's slightly uncomfortable, but you don't start shivering yet, and then after 2 hours, put that same person into the PET scan again, you can see now all the glucose is being uptaken by the cells, and that is because these fat cells are now activated, or as they call it, we have that browning effect through non-shivering thermogenesis. Unfortunately, overweight people and old people, if you do the same experiment, you can see there's barely any uptake. And then there was another study in 2012 that analyzed these fat depots, and actually said, oh, it's not true brown fat, it's actually beige fat. And they also show that beige fat can be shown all over the body in specific subcutaneous fat depots. So by activating beige and brown fat, people think there's like a therapeutic potential to treat obesity. And this discovery of beige and brown fat activation emerged a whole new field of fat metabolism. And the benefits of browning are increased metabolism, insulin sensitivity, and also energy expenditure. And there are several ways known how to activate brown fat. It's through cold exposure, as I showed you, but also exercise or endocrine and factors and metabolites. So basically, this whole field of research wants to turn a person that looks like that into a person that looks like that without any dieting or exercising, just by activation of brown fat. And that's obviously a huge market, or has a potential to be a huge market. So people are interested in drugs or other treatments like cold or energy devices. So this research is basically split in half. A lot of people try to find new therapeutics, and a couple have been found already, like I told you, like the cold treatment or dietary influences, like you can eat peppers or caffeine or green tea that is increasing your brown fat, but you also can exercise. And I want to focus on the diagnostic part to really see, okay, on a cellular level, what is happening in the cell? How can we see maybe a transformation of white into beige or brown fat cells, or is that even possible? And therefore, we wanted to have a high-resolution imaging platform that's ideally label-free, non-invasive, and non-destructive. And when I started my research in this field, there was not a lot that could do that. So we were looking into fluorescence lifetime imaging microscopy. And a lot of people talked about this already, so I won't go into detail of this slide. But basically you excite a fluorophore, and then you have a mono or multi-exponential decay of the fluorescence. The fluorophore we wanted to look at was NADH. And here's a little comic of the metabolic processes you see in a cell. And as you can see, NADH is highly involved in a lot of metabolic processes. NADH is fluorescent, its counterpart NAD plus is not. So that always shows that there are differences in fluorescence intensity, but also, as most of you probably know, NADH can be bound or free depending on where it is in the cell, and the environment can change the fluorescence lifetime drastically. NADH has this nicotinamide ring that is causing fluorescence. Also, NADPH has the same ring. However, luckily, for example, Melissa Scalar and all others showed in their papers that the NADH concentration is 4 to 40 times higher. So we basically neglected NADPH and just looked at NADH in our research. And here is something very traditional. It's the free to bound ratio, which is a ratio of glycolysis versus oxphos. As you can see here, this is a cell culture of adipocytes, fat cells, and then we put some component into the media of the cells to increase the metabolic activity. And as you can see here, the free to bound ratio is then decreasing, which means you have increased oxphos. And traditionally, the short lifetime component is associated with the free NADH and glycolysis, and the long component with the bound NADH and oxphos. And you can also do the same analysis with the phasor plot. And as you can see also, you can see changes. We looked into the literature, and when I started my PhD, I found these two papers and was interested in them because NADH in these two papers actually had 4 distinct fluorescence lifetimes. So I wanted to look into the fat cells, the adipocytes, and see if I can also maybe find 4 fluorescence lifetimes instead of just 2. So here's a typical image of adipocytes, and we did a 2-photon excitation, and then we analyzed a single pixel, did the fluorescence lifetime decay, and as you can see, we have, just like it used to be, we have 2 fluorescence lifetimes in that one pixel. However, if I now analyze all the pixels and do a histogram of all the lifetimes I find in my cell culture, we were very happy to see that we have 4 distinct fluorescence lifetimes, just like in the 2 papers I mentioned earlier. So we were thinking, why do we see 4 fluorescence lifetimes while other people only see 2 fluorescence lifetimes? And I think the reason might be because we have a higher effective resolution. What does that mean? When you do fluorescence lifetime imaging, to increase the photon count, you often do spatial binning. And a lot of people and a lot of publications I found use relatively high spatial binning of up to 9 pixels, or in this case was often more than 6 micrometers. And if you look here, this is a comic of a mitochondria. You basically get information from the mitochondrion, but also from the surrounding area, which might be the cytoplasm. And then it's all mixed together, and you have these 2 fluorescence lifetimes. However, if you have a very high spatial resolution, we only use 3 pixels and 2 microns. Often it's hard to get the right photon count, but if you do long measurements, we still get a decent photon count. And we think now we are a little bit more specific, that we have the fluorescence lifetime of mitochondria and the cytoplasm separated. And then we found 4 fluorescence lifetimes, and also the phasor plot was broadening. So we wanted to test our hypothesis. Is it really like that we have 4 fluorescence lifetimes, and where are they coming from? And what we looked into is we found that component 1 and 3 are mostly found in the mitochondria, while lifetime component of NADH 2 and 4 are mostly found in the cytoplasm. And if we now create that image, you can see here, this is all component 1 and 3, and this is the blue color is component 2 and 4. So based on the fluorescence lifetime of NADH, we can say, hey, this is a fluorescence from mitochondria or fluorescence from the cytoplasm. We want to be really sure. So the next step, what we did is we isolated mitochondria from our fat cells. As you can see here, we also did a mitochondrial stain. It's not perfect. For example, here's a nucleus also. But once we did the fluorescence lifetime imaging, we can see, yes, like we expected, component 1 and 3 is very prominent, which is what we expect to be the fluorescence lifetime from the mitochondria. So based on this, we created a new optical biomarker, and we called it the mitochondrial cytoplasmic ratio, or the MCR metric, which is showing the fluorescence intensity differences of mitochondria versus the cytoplasm, and it's the intensity. However, how we find the mitochondria and the cytoplasm is based on the fluorescence lifetime. To check if this new biomarker we found or created is really valid, we try to compare it to the gold standard to measure the metabolic activity, and that is an extracellular flux analyzer to measure the oxygen consumption rate, or OCR. Basically what that device is doing, it is measuring the oxygen quenching in the media right above a cell culture, and then based on that you can see, hey, that cell culture is using that and that amount of oxygen. And here you can see, usually you have a baseline, then you put in or inject into the media some metabolic inhibitors or stimulators, and then you can see the oxygen consumption rate is changing based on that. So we did exactly the same with our cell culture, and then compared it with the oxygen consumption rate. As you can see here, we have the baseline, then we inject an inhibitor, a stimulator, and another inhibitor. And you can see the color is changing. And also we look at our MCR value, and here we just inverted it, so you can see it's going the same trend as the oxygen consumption rate. For the FLIM images, we did individual injections, while with the seahorse analyzer or the extracellular flux analyzer, you usually do consecutive injections. And there's a good publication from Patrick Schaefer about this, explaining why. However, what we wanted to show is, and what we were very happy, that we have a very nice correlation of the MCR value with the oxygen consumption rate. And why do we need to have FLIM if we already have the extracellular flux analyzer and we can measure the oxygen consumption rate? One very neat thing about FLIM is you have that visual information. For example, here we can see we have a cell culture and we can look at 10 different cells, and each of these cells has a different free to bound ratio, but also a different MCR ratio, which you cannot do, for example, with the flux analyzer, because you only look at the oxygen consumption rate of the whole cell culture. So you cannot look at individual cells. And here we wanted to show that the MCR metric has a higher dynamic range compared to the free to bound ratio. So this was all leading up to that one experiment we wanted to do. We are interested in brown fat. So now we have the method, we have the MCR ratio. So what we wanted to do is we wanted to turn a white fat cell culture into a beige fat cell culture. And there are several drugs that are already known that can do that. One of them is epinephrine, and the other one is forskolin, and they are known to induce browning. And then we did that, waited for 4 hours, and measured the MCR, the oxygen consumption rate, and the uncoupling protein 1, which can only be found in beige fat cells or bright fat cells and not in white fat cells. And as you can see here in this Western blot, epinephrine and forskolin definitely induced that UCP1 expression, which you can also see in this graph here. It's significant, while the housekeeping gene stays the same. If we look at our flux analyzer, the red bar, we can also see that the oxygen consumption after 4 hours is significantly increased, and also our metabolic ratio is also increased significantly. However, the free to bound ratio, we expect it goes down here, which it did not do in a significant way. So we were very happy with this result, that we now have a metabolic biomarker that might have a higher sensitivity than the free to bound ratio. And I showed you this graph before. It's we looked at the MCR values in relation to the oxygen consumption, and we did that for adipocytes. But we were also thinking, hey, what if we could use that same ratio for other cell types? So we looked into HEK cells, which are human embryonic kidney cells, and we were very happy to see that same method also works for this type of cell. So then we were excited and say, hey, maybe we don't even need the flux analyzer anymore. We just look at the MCR value and we know exactly how much oxygen a cell is consuming. Unfortunately, that wasn't the case, because it really depends on what cell culture you're looking at. And here I just compared the adipocytes and the kidney cells, and as you can see, if we just pick an MCR value, it does not correlate to the same oxygen consumption rate for either cell culture. So we think you can use this biomarker as a relative measure, and or maybe in combination with a flux analyzer. But as of now, you're not able to see absolute values, just relative values, how your cell culture is doing, increasing their metabolism, or if it's inhibited. Another really neat thing of having this visual information is that you can analyze mixed cell cultures, which is very difficult with the flux analyzer. You can look at the overall oxygen consumption rate of a mixed cell culture, but obviously each part of the cell culture will have different metabolic activities, and that you can easily distinguish with FLIM. And we can look at our metabolic ratio, and we can see it for both the fibroblasts and adipocytes, but we cannot see that, for example, with the flux analyzer, which is a big advantage. And with that, I just wanted to summarize. We are able to see 4 distinct fluorescence lifetimes in adipocytes. Component 1 and 3 were associated with mitochondria, and 2 and 4 with the cytoplasmic fluorescence. We created that MCR metric which has a higher sensitivity than the free to bound ratio. And we were actually, what we wanted to do, able to measure the browning of white adipocytes. And the MCR value correlated nicely with the oxygen consumption rate. And for the conclusion and the outlook, we think FLIM is very powerful label-free tool, and the metabolic activity can be used, for example, for flow cytometry, as earlier mentioned, and very easy to monitor metabolic changes. And we think you can go beyond fat, so you also use it for other fat cells, but especially for the fat and the browning, it might be interesting for drug development or screening, flow cytometry and cell sorting, all these kinds of things. And yeah, the absolute goal would be having an absolute quantification, but we are not there yet. And with that, I would like to thank everyone, especially my PI Dieter and my PhD advisor, and thanks for your attention.
All right, thank you for the talk. We have a few questions in the chat for you. The first one is, are you sure that the fluorophores and lipid droplets are associated to NADH? It looks like they're a deviatic metabolic trajectory of free to bound NADH in the phasor plot.
So about the lipid droplets, there have been a couple papers that show that the lipid droplet itself has a fluorescence lifetime in that range. So what we did, we always excluded the lipid droplets from our analysis. Here's one slide how we did that. We used SIS and CARS to really show where the lipid droplets are in the cell. And then we created a mask to remove the lipid droplets from our analysis. And as you can see, even with the lipid droplets removed, we still have 4 distinct fluorescence lifetimes. So we are pretty confident that it was not the lipid droplet fluorescence that gave these additional fluorescence lifetimes.
Okay, and do you see a different lifetime signature in different types of fats?
So yeah, we looked into that, and basically we had stem cells, and we took those stem cells out of white fat depots, out of brown fat depots, and out of beige fat depots. And then these precursor cells we differentiated into fat cells, and we were able to see different fluorescence lifetimes for all 3 differentiated fat cells. Yeah, we think just based on their also how the fat cells look like, you can see clearly differences in the fluorescence lifetime.
Okay, and then the fluorescent lifetimes for NADH, and what do they correspond to?
Can you repeat that, please?
What are 4 fluorescent lifetimes for NADH, and what do they correspond to?
So yeah, as showed in the talk, we think like the component 1 and 3 are probably the fluorescence lifetimes for the mitochondria, and component 2 and 4 are mostly found in the cytoplasm. What exactly is causing these fluorescence lifetimes, we don't know yet. We have to analyze that. It might be that one is like, it might be a conformation change, or the environment that the mitochondria is creating or the cytoplasm is creating, like pH changes. We really have to dig deeper to find what exactly is causing the 4 fluorescence lifetimes.
All right. So there's a couple more questions in the chat. I'll let you get to after the after your talk, but I think we need to move on now. Thank you very much, Michael.
Okay.